Last recordings. Yeah. Did we ask, it's okay to record, right, Eric? Before? Okay. Yeah, please get connected. Thank you.
Lovely. All right, everybody. Today we are here with Eric Sun. He is a final year PhD student in biomedical data science program at Stanford University, where he is jointly advised by Dr. James Zhao and Dr. Ann Burnett. Previously, he earned a bachelor's degree in chemistry and physics and graduated summa cum laude from Harvard University. And his current research focuses on designing and deploying machine learning models for the analysis of single cell and spatial omics, particularly in the context of aging. Today, he's going to talk about this idea of whole transcriptomic spatial profiling of genes at a single cell resolution and how it remains a challenge. And to address this limitation, spatial gene expression prediction methods have been developed to infer the spatial expression of unmeasured transcripts, but the quality of these predictions can vary greatly. So he is going to present this transcript imputation with spatial single cell uncertainty estimation, also known as tissue, as a general framework for estimating uncertainty for spatial gene expression predictions and providing uncertainty-aware methods for downstream inference. And with that, I'll hand it over to Eric Sun.
Cool. Thank you for that introduction, Hector. So yeah, as you described, I'll talk a bit about our project building tissue, so some of the benchmarks, how the method works. And then I'll kind of switch gears towards the end to talk about application that we've been kind of excited about using this tool to learn a bit more about the biology of brain aging. So yeah, so I think to start off, I will kind of want to give just a brief overview of like two flavors of single cell transcriptomics that are kind of important to the main thrust of this project, which is like one is dissociated single cell transcriptomics. This is like a pretty, I think, well-established tool at this point in time. And basically what you do is you would take, a given sample, you would dissociate the individual cells, so you lose the spatial context. And then using certain microfluidic technologies, you're able to, barcode individual cells and then measure gene expression in individual cells. And this has been like kind of widely applied across multiple fields of biology and medicine at this point. But kind of more recently, people have moved towards spatial technologies, and particularly like these imaging-based spatial transcriptomics technologies. And for these, the technologies are quite different. It's imaging-based, so they usually use some kind of fluorescent probes that hybridize to mRNA transcripts that they want to target specifically, and then do sequential rounds of imaging to recover the locations of individual transcripts in the tissue. So you preserve the spatial location, but there's some trade-offs which I'll talk about in a bit. And these kind of, I think an easy way to think about these two different types of single cell transcriptomics data sets is to think of the dimensions of what they look like. So dissociated single cell RNA-seq data sets are generally, up to whole transcriptome, so like 10,000 to roughly 20,000 genes that you can cover in one experiment. And then the number of cells varies quite a bit, but to think of it, generally, let's say you have 10,000 cells for a given type of sample. You have a different order of magnitude when you do like an imaging-based experiment. And so for that spatial experiment, you'll get something like, maybe a few hundred at most genes that you can target. So these are usually pre-designed panels. So like dissociated single-cell RNA-seq, you can kind of profile the entire transcriptome in an unbiased manner. And spatial transcriptomics, because you're designing these individual probes for a lot of these imaging-based approaches, like you can only pick like a certain set of genes. So it only spans maybe, up to maybe 10% of the transcriptome at most. But you do get profiling of a lot more cells because you keep the tissue intact, so there's not a lot of loss of cells from dissociating tissue and then filtering the cells. You do get to image most of the cells within a tissue. And so you get maybe an order of magnitude more cells, but at the same time, less genes. So there's kind of this trade-off between the two different types of approaches. And so the question that kind of motivated our project building tissue is like, can we get the best of both flavors of single cell transcriptomics? Can we both preserve the whole transcriptome kind of information, dissociate a single cell RNA-seq, and then also have the spatial context and the greater number of cells that's present in imaging-based spatial transcriptomics. And a few methods have been already developed for addressing this kind of integration. And so people have developed, for instance, like one flavor of methods which do some kind of transport of the single cell RNA-seq, so each cell with its entire transcriptome, and transport it onto a kind of spatial coordinate set. So they usually use like some kind of optimal transport framework. Conversely, there's a different approach where maybe you want to preserve the exact spatial coordinates of the spatial transcriptomic cells, and then you ask to impute basically the missing genes. So maybe you have 100 genes in the spatial transcriptomics, and for each of those cells, you impute the missing 19,900 genes using a kind of a reference single cell RNA-seq data set. And so this is the type of the class of methods that I'll mostly talk about today, because at least for a lot of the projects I've worked on, preserving exact kind of cell spatial proximity differences is pretty important in terms of knowing which cell is exactly next to another cell. And so this type of approach tends to kind of favor that and over, for instance, like the quality of or accuracy of the exact kind of gene expression measurements. And so even for that class, there's a lot of methods that people have developed. So this is like a recent benchmarking paper where they looked at 8 different methods. I think by now there's probably at least 20 different methods that do this kind of imputation approach. And you can see there's variability in like performance of these different methods across different benchmarks. Here is just like a Pearson correlation over the gene expression predictions for each gene. Kind of zooming out one class of method, which I think is kind of representative of the majority of these methods, is that they generally approach these imputation tasks as kind of a joint embedding and nearest neighbors search. So what you do is you have your single cell RNA-seq data set, which is like the square. You have the kind of the more narrow matrix from the spatial transcriptomics data set. You do some kind of joint embedding, so you get them into the same embedding space. And then you do like a nearest neighbor search for each spatial cell. You ask, where are the 10 nearest neighbors in the single cell RNA-seq data set, and then you average the gene expression to impute the missing gene expression for the spatial cell. And so a lot of these like methods kind of fall under that kind of branch. There's a few other ones where they use some different methods and so forth, but generally like the idea is you collapse the 2 data sets and then use that to infer the missing gene expression. So one thing we noticed, like both in that benchmarking paper and when we implemented and applied some of these methods to our own data, is that there's a lot of variability in the quality imputation. So there's a lot of variability in terms of which methods perform the best. There's no clear method, so you use different metrics here. I've shown like Spearman correlation, so how well the order information is preserved across different cells for a given gene. If you look at absolute error, there's a difference, like the order shifts a bit. So depending on which metric you use, which data set you look at, which method performs the best is not really clear. And then even across different data sets, which you can see here. So each set of violin plots for a different data set, a different tissue, and a different spatial kind of imaging-based technology. There's high variability across data. So some of them perform really poorly across most of the methods. Some perform well for certain methods, but bad for others. Some of them perform well for most of the methods. And then, so there's that kind of inter-data set, inter-context kind of variability. There's also a lot of variability even after fixing the data set and the methods. So for instance, like you can look at, the violin plots here. There's a lot of spread in terms of like, even within a given data set and a method. For some cells, some genes, you get really good prediction, and some cells and genes, you get pretty bad prediction. And this is, I think, incredibly important because for people who want to use these imputation tools to then do downstream analysis to generate hypotheses that they can bring to the lab to test, it's really difficult to trust these imputations in those types of downstream analysis. Like, maybe some of the high performing genes, like here, maybe a correlation of 0.8 is good enough, but if it's like 0.2, 0.4, maybe that's not quite good enough for getting a kind of highly confident downstream interpretation. So the idea is like, can we build in like a measure of this uncertainty and then use that to kind of improve or make these kind of downstream analysis that rely on imputations, make them more robust so we don't have so many false positives. And so this is where our method comes in. We call it tissue. But basically it's estimating uncertainty for these like spatial gene expression imputation methods. So it's a decently complex pipeline. So I'll kind of step through it kind of step by step. So like generally first you start with like this spatial data. So there's a set of measured genes, maybe let's say 100 genes that are measured. You have a paired kind of RNA-seq data set that would be usually from the same species, same tissue. And then you do this like black box gene expression prediction, which can be using any of those imputation methods I discussed previously. And we do this in kind of a cross-validated setup where you hold out a few of these measured genes, and then you make prediction on the held out measured genes and on these like genes you actually want to predict. So the idea is that you build this calibration set here where you know the true measured gene expression, but you also know how well the model is either over predicting or under predicting on those held out genes. And then that gives you a way of calibrating uncertainties later on for the predicted genes that you actually care about. And then from these predictions, this is a kind of like a more technical detail, but we found that if you do stratified groupings with certain genes, certain cells have different types of performance behaviors under different imputation methods. So if you do this kind of stratified grouping and build calibration sets separately for different groups of genes and cells, you get kind of improvements in performance, especially for edge cases where if you do have a kind of a blanket approach, it doesn't quite work for some of these like more rare kind of behavior groups. At the same time, you also build these like spatial neighborhoods. So the idea is like you want some kind of measurement of uncertainty that's agnostic from the actual measured gene expression, because you don't have that for the predicted ones that you actually want to impute. And so for this, we developed this like measure we call this cell-centric variability, which is the idea that for cells in close spatial proximity to each other that share very similar gene expression, they will probably more than likely share similar gene expression for genes that weren't measured. So the idea is that if your cell is the same cell type and you're very close to another cell of the same cell type that shares very similar gene expression profile for 100 genes, it's very likely that for the other 19,000 genes you'll share very similar gene expression. And so we use that as a way of calibrating or measuring this like rough uncertainty, which is like if there's a lot of variability. Despite that, then maybe the prediction is not quite great, but if there's a lot of stability in that kind of the gene expression of those similar cells, then maybe the prediction is pretty good. And we use that along with the kind of held out calibration genes to calibrate uncertainty. So this comes in from a few of the equations, which I won't dig into too much unless there's questions later. I have a slide where we have these equations. We can talk about it more then, but basically that's kind of like the idea of what we're trying to achieve here. And then finally, basically, you build this distributions for both the upper and lower bounds for these confidence intervals. So the final output is basically we get these uncertainty measurements, we build these calibration scores, and then we use these calibration scores to design a confidence interval. So for a given confidence level, you can say maybe I want a 95% confidence interval or prediction interval for these imputed genes. Tissue calculates like basically an interval around the predicted gene expression. So say with 95% confidence, maybe the actual gene expression will be between x1 and x2. And we do this separately for lower and upper bounds because gene expression just is not very normally distributed, which is generally what people in this conformal inference field deal with. But it's very there's a floor at 0, and then there's a very kind of fat tail for positive gene counts, and so that's why we do the two separate kind of intervals there. Cool. And so we want to build these confidence intervals. So what we do is then we calculate these calibration scores, and it's relatively easy to go from that to the confidence interval, which is basically we want maybe 1 minus alpha coverage, alpha being some kind of error rate that you specify. Then you can just basically take that quantile in the upper confidence interval calibration score distribution, the same in the lower kind of interval distribution, and then you multiply that calibration score quantile by just the UIJ here, which is like the cell-centric variability I talked about, which is like a unique kind of measure of uncertainty for each individual cell, and that will give you an exact kind of confidence interval for each cell and each gene. So each basically prediction value gets a kind of a unique confidence interval. And so what do these confidence intervals look like? So you have like, for instance, like I showed kind of 3 different examples here. On the left is like the actual imputation error. So there's like if we hold out a few genes, we make imputations using the remaining genes, and then look at how much error there was between the held out kind of ground truth and the predicted or imputed gene expression. And on the right, we have these the width of the tissue interval. So this is like during the tissue process, we construct these prediction intervals, and then plot the width of these intervals. And you see, like, visually they generally match up. There's like maybe a very slight discrepancy in a lot of cases, but like generally where there's like very high prediction error, there's very high or wide kind of a tissue prediction width. And there's like the three different methods, three different use cases across, I think brain tissues on the left and right, and like a Drosophila embryo in the middle. And so, of course, like we want to look at this systematically. I think it's always good to get some appreciation of what, how it looks visually, but we wanted to look at this across, 11 different data sets. So using some data sets from this previous benchmarking paper and some that we kind of collected ourselves, like we have, 11 different data sets, about 4 different species represented. There's mostly brain and embryo. So there is a bias, I think, in spatial biology right now for maybe certain tissue types over others, and so I think that's kind of reflected in our benchmark data set. So that is definitely some limitation. We're definitely interested in seeing how it performs generally across other contexts. But even with this, there's 8 different spatial technologies represented. They're paired with single-cell RNA-seq data from roughly 3 different technologies. So there's like 10x Chromium, SmartSeq, and then I think a few of these other one parse bios like technologies. So I think there's decent diversity here in terms of like potential use cases, and you can see across most of these cases there's pretty good calibration. So these are calibration curve plots. So how to read them is like on the x-axis you have like the tissue coverage. So this is like the theoretical coverage. So you give tissue an alpha, and you say I want 1 minus alpha coverage. So maybe you give alpha error rate of 0.05, and you get 95% coverage. If we do this for every value of alpha. And so you can map those out, and then for this, at the same time, you can count up the number of actual like measure values or ground truth values that fall within that prediction interval. So the idea is exactly 1-to-1, which is meaning that the tissue interval is exactly accurate to how it should be defined. You get a straight line, basically, y equals x across this plot. And you see that's generally the case across most methods and most data sets. In some cases, you get a little bit of a deviation. I think a lot of times those are for the data sets that have a lot more genes in the spatial gene expression data sets, so a lot more variability and like a lot more dropouts, higher sparsity, and maybe less confidence in the actual measured expression values. But I think generally across most cases you see decent calibration. Cool. And then so I think from the few experiments we've done, we think that tissue can provide these calibrated measures for imputation and uncertainty for spatial transcriptomics. But I think the big question is like, how is this useful? Like it's, even if you get a sense of uncertainty measurement, for a lot of people who want to work with these data sets, it's not super clear how getting a prediction interval or getting a measure of uncertainty, even if it's accurate, how that can improve any type of downstream analysis or make their interpretations more robust. And so this is kind of like the latter half of this project that we're really focused on, which is can we use tissue to uncertainty to kind of improve downstream data analysis. And so this required building a lot of kind of more custom frameworks that will leverage uncertainty, because there's not currently, generally all these downstream tools, just their input is basically a gene expression matrix without any measure of like uncertainty in the measurement or uncertainty in the imputation. And so we developed a few other tools that kind of leverage that information in addition to the actual predicted gene expression to hopefully improve kind of some of these analyses. And so like one example I want to highlight is like using tissue for hypothesis testing. So this is like a very common approach in bioinformatics and pretty much all biology at this point, which is like if you do any kind of unbiased or even biased kind of omics type of approach, you usually get a lot of different readouts. So in this case, genes, but you can also think of metabolized proteins, a lot of other types of molecular species that people would then want to say, look at systematically which ones are the ones that are actually contribute to certain processes or different protein conditions. And so this generally relies on like statistical hypothesis testing. And the way we kind of adapt this uncertainty approach to your kind of traditional hypothesis testing is to building these uncertainties by generating multiple imputations. So the idea is that you have one prediction, right? So this is like the orange matrix that's shown on the left, where you have the predicted gene expression. The idea is you can generate multiple imputations for individual by just sampling from the calibration scores from tissue. So if you sample a calibration score for each of these individual cells, you can update that using kind of reverse basic engineering the tissue prediction interval, and then generate basically D multiple imputations. So like just that represent kind of the distribution of these calibration scores. And the idea from there is that then there's a lot of kind of well-built theories in multiple imputation that you can leverage to build these kind of rigorous statistics from, if you have like, 10 roughly independent imputations, there's a way of combining them to get a more robust kind of p-value readout. And so the idea here is that we use that and then compute calibration score, sample, and then leverage this to perform the statistical tests. So we've kind of applying this method across a few different data sets. So these are examples where we I'll talk a little bit about the scheme where how we do this analysis, but basically there's certain conditions like cell types or anatomic regions that we expect that there should be kind of true differences in gene expression. So these could be cell type markers, or just anatomic regions have maybe different markers. And so we built a ground truth of basically comparing the measured genes. So we you have, let's say, 200 genes in a data set. We perform regular differential gene expression analysis at different levels of confidence, so at different p-value cutoffs you can think of. And then this serves as kind of the ground truth, like positive versus negative kind of detections. And then at the same time, we do the approach with the baseline imputation. So if you were just to impute those genes instead, and then perform differential gene expression analysis using that same setup, what is your false positive or false discovery rate? And then what happens if you use tissue to do that imputation, you leverage that uncertainty with a multiple imputation approach, what then is that false discovery rate for these different p-value cutoffs? And so you can see here for across each of these different contexts on the each row is a different kind of gene, spatial gene expression imputation method, and then each column is a different data set. And across, the x-axis is basically this number of discoveries, which is like a tunable by how much, by where you cut off the p-values, basically for determining if something significant or not significant, differentially expressed. And then you can see like the red curve here is the baseline approach, and then tissue generally has across basically the entire range of possible number of discoveries you want, I think pretty substantial reductions in the false discovery rate. So I think we found that quite promising in terms of at least leading to more robust kind of discoveries and using computer gene expression analyses. Yeah, and then we've also looked at a few other applications. So aside from differential gene expression, of course, there's like a lot of other applications that people use these types of single-cell transcriptomics to perform. And so kind of broadly, some of these are like you maybe train some supervised learning models. This could be a deep learning model, it could be just a regression model, it could be a classifier. People are very interested in clustering their cells, into different groups and trying to annotate what each of these clusters mean. They're also interested in visualizing their individual their data sets. So this is your UMAP, TSNE, PCA visualizations. And so these are kind of three kind of, I think, broad application areas that we wanted to test out too. And so we have a kind of a unified approach for each of these cases, which is that we compute these tissue prediction intervals, where these are these asymmetric kind of prediction intervals that cover roughly where we think the true measured gene expression should be at a certain confidence level. And then you can measure the prediction interval width as a kind of a correlate for how confident the prediction is at that point, and then do some filtering. So the idea is that you can remove cells with really high uncertainty in a gene specific and class specific manner. So once you normalize across gene expression level or the gene itself, like there might be some cells that have really poor predictions with really high uncertainty. And for a lot of those cases, maybe you just don't want to use them in these downstream analysis. The idea is to remove a small amount of these kind of high uncertainty cells, and then using that kind of filtered out data set, perform these same types of analyses. And in a lot of these cases, like you would think that with a smaller data set, especially when, like supervised learning or something, you would have decreased performance. But even in those contexts, I think we see mostly improvements. And so, this is just one visualization that summarizes most of these experiments that we did. So on the x-axis are a few different metrics. So you have a few things like, accuracy, F1, and the ROC, AUC, which are kind of these classification metrics for if we build classifiers for each of these data sets. So on the y-axis is a given data set and the type of label that we're either clustering by or training a classifier to predict. So the cell type, it could be anatomic region. And then you can see here, everything in green is, so the colors are this, it's like a log fold change and improvement basically over the baseline approach, which is like if you just, throw all the data into the model instead of this filtered down data set using the tissue uncertainties. And you can see like everything in green is like a slight improvement. Everything that has a star is something that's statistically significant improvement after doing, multiple hypothesis correction. And so I think, most of these cases you see decent improvements. Similarly, if you look at clustering as a downstream analysis, so the idea here is that we perform like a k-means clustering and then ask like, what is the adjusted RAND index, or what is the agreement and cluster assignments between the ground truth, which is like the actual label, and like the clustering approach. And you can see here that, generally there's improvement, so a lot of green. And then similarly for separability, so the idea here is that, it's hard to exactly assess like what is a good visualization, what is not in terms of the data. So the idea here is we project the data down to like a very low dimensional space, so akin to visualization, then ask like how well can we separate this using like a support vector classifier linearly. And so the idea is that you get good separation across these different labels, you have a decent visualization that tends to separate out important clusters. So we use that metric here, and you can see again, like generally if you use tissue to filter down the data set first, you get pretty robust improvements. Yeah, and then I think the final vignette I go into, and I'll take a little bit of question after this before I transition to a more a different story. But I think one of the use cases we want to highlight was like, a lot of this project was motivated early on by a MERFISH data set we collected. So MERFISH is one of these imaging-based spatial transcriptomics approaches, and so we were interested in brain aging, and we particularly in this region of the brain that is resident to neurostem cells. So these are in the lateral ventricles, these are cells that can then proliferate and give rise to neurons throughout life. So they're very important. Like when you think about how to rejuvenate the brain, how does the brain respond to injury throughout life, and they're known to kind of decrease in function during aging. So they're very kind of interesting cell type in that context. They also have a lot of these kind of substates. So you have these neurostem cells that are quiescent, and then they can switch into being activated. So generally with aging, you get a higher proportion of quiescent neurostem cells. So these are less likely to proliferate and give rise to new neurons, and a decreased proportion of these kind of active ones. And, the active ones can then proliferate and give rise to neuroblasts, which then are these kind of intermediate cells that then give rise to new neurons. And so even within this like lineage of cells, there's a lot of kind of important distinctions between different substates. And so that kind of question was, can we measure or identify these cells using spatial transcriptomics in this region? So what we did for this is we had, I think in this case, just a single mouse, a young mouse. We took out, a coronal section of the brain. We looked at, we performed MERFISH for 140 genes, and then looked at the areas right around these lateral ventricles where these cells are located, and then just annotated them based off some of the markers we included. So, in designing these gene panels, for these particular cell types, they haven't ever been profiled using any MERFISH or other types of technologies. A lot of times it's like trying to, cobble together some, genes that correlate with the protein markers that are generally used in the field, or, genes that come out from like single cell RNA-seq data sets. But a lot of times these aren't exactly, 100% reliable in like a MERFISH context, for instance. And this is what we found out is like you cluster the cells, but then as you can see, like in the cell type list, there's no neurostem cells, there's no neuroblasts, there's no quiescent activating neurostem cells. And a lot of these fall, I think, under this blue cluster that we call ambiguous. So if you zoom in on the lateral ventricles, you can see they lie roughly where the neurostem cells should be. But we weren't able, with the markers we picked, able to distinguish between these three kind of important distinct subtypes of this ambiguous cluster. And you can see like they like, physically they are where they should be, but with the gene set that we picked, it wasn't possible to disentangle that. And so of course we ask the question, like maybe we could use this method we developed to annotate subtypes in this cluster. To do that, basically we use like the hypothesis testing approach I outlined previously, which is you can perform differential gene expression analysis at whole transcriptome level by pairing this MERFISH data set we collected with previous data sets using single cell RNA-seq. And then so by doing that, we can basically augment the number of markers we're using in the spatial data set effectively. So here on the x-axis are all these different markers we added. Everything in purple are for like these quiescent neurostem cells. Everything in this like blue color are for activated neurostem cells, and then for neuroblasts, which are kind of the more downstream members of this lineage, we have these like orange labels. And you see that here, using this tissue T statistic for the hypothesis test, you can see that for certain classes of genes, there's like upregulation, significant upregulation, and then significant downregulation relative within this ambiguous cluster for other genes belonging to other cell types. And using this approach, you can pretty reliably annotate like neuroblasts should be like the first cell type, and then then activated neurostem cells and quiescent neurostem cells. And so we kind of go from this very bulky kind of ambiguous cell cluster to a more refined annotation in individual cell types that we're actually interested in. So I think the idea here is to demonstrate like one potential use case that we found kind of useful in our own research was basically using this tool to recuperate some quote unquote failed experiments, where a lot of times when you're trying to identify a particular cell substate or subtype that doesn't have a well-known cell type marker panel that's been using these imaging-based technologies, like you do run the risk of maybe not including enough markers, not including the right markers. And a lot of times I think using this as like a way of recuperating that signal that might have been lost from gene panel design. Yeah, so I figured I could stop here a little bit. I'll talk about, I think for the last half, I want to talk a bit more about kind of recent project that we've been working on where tissue has been kind of useful. But if there's any questions like about the tissue methodology, some of the benchmarks we looked at, I figured this would be a good time to take those questions.
So Eric, quick question. How do you validate your findings? Because it's very easy to use any classifier to classify into A and B. Most humans, most cells tend to be a continuous spectrum. Nothing is absolute in biology. So, or most things are not absolute in biology. Let me rephrase that. How do you validate?
Yeah, no, that's a great question. I think it's hard for each individual case. I think in some cases, so there's another example I didn't present, but is in the manuscript where we looked at, for instance, granule cells in the dentate gyrus of the hippocampus. And we did a very similar approach where we looked at a lot of these different neuronal subtypes and asked, tissue to impute the expression of these markers, and then use that imputation to do hypothesis testing and annotate the right cell types. In those cases, you can usually verify that by the spatial location of the cells. So for instance, granule cells lie in a very particular part of the brain, and you use that as kind of a ground truth to annotate this. In the case I presented here, it's a little bit harder to validate because these cells are kind of heterogeneously located within that area of the brain. I guess the idea is that we have a few we've collected new data sets, let's say, which I'll present on, but with more genes and stuff, and these have similar distributions in terms of where the cells are located, which markers are lighting up for these individual cell types. But that's,
Is there a minimal set of markers that you use to annotate a single cell?
Yeah, so I think in some cases you can probably annotate some cell types with one or two markers, but these are ones that people have shown that, for instance, a ligand sites, you can maybe do all two, and that would be enough to annotate them. But I think it's confirmed mostly because they're known to be in the white matter tracks. You can look at brain sections, you can see that they're generally labeling the right cells. And a lot of people have, validated this across other MERFISH data. I think it's a little bit harder. So in these cases, we included maybe 5 or 6 different markers for nerve stem cells, and even that wasn't sufficient. Some of them may be a little bit too lowly expressed or something. It just, I think, it takes a bit of trial and error. But I think now that we've done this a few times, we have a much better idea. But yeah, it's a little bit difficult, I think.
Thanks.
So is that the end of the talk, Eric?
No, he's just taking a break and is
Okay.
going to continue on to aging now.
Aging, okay, because I just want to make sure I need to help or not. Okay.
Yeah. All right. So I'll move on to kind of this a newer story, which usually comes into play a bit, but just kind of set up like the background for this. A lot of tissues, so particularly the brain, are very complicated in terms of their spatial organization. So you can see here a lot of different spatial regions. A lot of them have been annotated for different types of cognitive or behavior functions. And they all kind of interact together. Individual cells, have projections. There's a lot of important circuits in the brain. And of course, all this should contribute or is likely to be involved in both aging or be impacted by aging across the life of an animal. And so the question we were kind of interested in answering was, can we use spatial transcriptomics to understand cell proximities that drive aging? Because a lot of current brain studies at single cell resolution generally do dissociated approaches where you take very spatially rich kind of organized brain tissue, you break it apart and you lose that spatial information. So you only look at changes within individual cells. So can we keep all the spatial information intact and use that to then understand how cells maybe influence each other during the aging process? And so to answer that question, we collected a pretty large profile. So this is like kind of the second iteration, where the previous MERFISH, we did 140 genes, kind of a pilot. It was enough to detect nerve stem cells. We've since kind of annotated more carefully, collected a larger gene panel. And so this is about 300 genes where we did a pretty wide range of ages. So this is really from early adulthood, so 3.4 months, maybe equivalent to roughly 20 years in human age, up to 34.5 months, which human equivalent years would be like 95 or so. So this is basically the entire kind of span of aging in mammalian animals. And so we profile here coronal sections of the brain and sagittal sections across a few other ages to get coverage of a lot of different types of brain regions. And so we use MERFISH. A few reasons we chose that technology is one, like we used it before, but I think generally it has a larger imaging field of view than some of these other methods. So you can get whole coronal sections and whole sagittal sections. So in total, there's roughly about 4 million cells across this aging axis that I presented, but also across a few other interventions that we looked at, like exercise and reprogramming, which are things that people have shown to maybe have promise in rejuvenating the brain that we wanted to see in that context. This is generally what the data set would look like for a MERFISH data set. So you have a lot of genes. So on the left have the UMAPs with these different sections, so a lot of different cell types, so 18 different ones, different neuronal subtypes, a lot of glia cells, and then you have on the right these kind of spatial visualizations. You see different regions light up. They're colored by individual cell type. You can even from that you can see like there's a cerebellum is very rich with their molecular granular layers. You have like the white matter tracts that really the oligodendrocyte rich and so forth. And so to kind of assess brain aging, we posted a type of modeling called building like aging clocks. And so this is something in the aging field where basically you train these penalized regression models where you fit them on gene expression or some other types of features. Generally, people have done like methylation or some type of epigenetic markers. And then you ask to predict, the model predict chronological age. And of course, there's going to be a lot of prediction error. And so the idea is that you get some kind of gene expression from that. So you train and evaluate these like that. They're useful for a lot of different things, but we were interested in asking, how do they influence aging in the cells? And so we, perform this matching analysis. And then from that, we identify that, T cells are the most pro-aging cell type using these spatial clocks, and nerve stem cells are the most pro-rejuvenating. And then we asked, what mechanism might mediate these proximity effects? And then that's where tissue comes in to measure, to understand which are the potential pathways mediating these proximity effects. We use that to impute up to 12,000 different genes, perform this differential gene expression analysis and pathway enrichment. And I'll summarize this relatively quickly, but generally we see interferon responses going up next to these T cells, and then fatty acid oxidation going up near these nerve stem cells using this tissue approach. And so using tissue and the kind of the spatial data set that we collected, we're able to kind of build this understanding of potential cell proximity effects that are important in governing kind of brain interactions. So the key takeaway is spatial gene expression imputation is a very powerful tool. Uncertainty kind of enhances the confidence that we can use these imputations for. Tissue does this in a calibrated manner that can improve kind of downstream analysis. And then we've used this kind of to uncover some mediators that we've also validated then using immunostaining and other experimental approaches that we think might help contribute to brain aging. Yeah, so I just want to thank all the important contributors, James, and my co-advisors, Rowan, Olivia, Lucy, Paloma, for their contribution to various aspects of this project and my funding sources. Cool.
Thank you, Eric, for the wonderful presentation from technology to aging. So are there any questions? Please feel free to directly unmute yourself or post on the chat, please.
Let's say my battery is getting kind of low, so if I cut out, maybe I'll give me like 1 or 2 minutes, and I can try to get back on.
So as we're waiting, I think the aging part, right? So there's the multicellular organism changes, right, in the aging, and there's those anatomical changes, right? And there's additional complicated but molecular, programs. So could you tell us a little bit about how we can maybe untangle, complexity of aging with your approaches?
Yeah, so I think the importance of these approaches is really bridging it from single cell differences. So those are like, you think of it like very linear kind of characterizations of aging to cell interaction level approaches, which is bringing up like one level of complexity where we have certain cells might be aging on a different trajectory either near other cells. And then, so that's like one level. And then in this project, which I haven't presented today, we've also done things where we try to be into a neighborhood level. So we use some like graph deep learning approaches to ask like then at a neighborhood level, if you were to ablate certain cell types, how does that influence aging? And so that kind of goes into much more non-linear, kind of more highly complex interaction. So yeah, so I think spatial data sets give you that information to really build off of. I think if you were to only work with single cell RNA-seq, it's much harder to think about how communities or different microenvironments might contribute to aging. So I think it's incredibly important to keep that kind of spatial biology intact in studying aging.
Exactly, thank you. And also I'm waiting, maybe Sunil Yashim, feel free to ask questions as well, of course. The first part, yeah, you have a question, Yashim?
No, I mean, the aging part is very interesting because you can apply these models exactly to the this is only for the mouse different months, but for the disease revealing, like I don't know, it's either brain or any other diseases that is aging important, since I'm looking everything from the cancer perspective. So I mean, this type of technology is very useful, and then using your methods definitely, as we see that very correcting the existing technologies, which we started using them, but they are looks like from the algorithm wise in their infancy. Thanks to your presentation today, we kind of started being aware of it. Thank you. It was a great presentation.
Cool. Thank you. Yeah, I agree. I think the disease is a really interesting context, and we tried looking at a few like mouse models of disease, and I think there's won't go into detail, but I think there's definitely interesting spatial insights that you can gain with these tools.
And also, oh, we have a question from Takashi, right? And did we lose Eric? I think we lost Eric today.
His question is, in aged mice, could you observe any degenerative regions? What is the cause of T cell activation?
Yes, but do we have Eric who can answer this question? I think he just dropped. Let's wait for a minute, maybe. Sorry, guys, for the technical difficulties. If not, we're gonna end here. And in the meantime, in 2 weeks, we have another speaker from UC Irvine, and also talking about another Nature Methods paper on inferring parent-driving intercellular flows from single-cell and spatial transcriptomics data. His name is Axel Ahmed from Professor Ching Ni's group from the Department of Mathematics. So they actually have been leading the cell communication, as you know, from the UC Irvine. So we'll be learning about this more next week. But this week, I think we lost Eric for now for the technical problems. And I don't see him coming back still. Okay. Then with this, maybe let's thank everyone. Let's thank the speaker again. Thank you everyone for coming. And let's celebrate at least Eric here. And we'll be sending him a note about your contribution as well. And with this, yeah, we're going to be wrapping up here. And thank you everyone for coming. Again, next week, we're going to have Dr. Axel Ahmed from UC Irvine talking about cell communication from Professor Ching Ni's group. And have a nice weekend and hope to see you guys in 2 weeks. Bye-bye for now.